we will barrel onto adaptive immunity then. So we've now covered many of these cell types. We'll come back to some of these granulocytes that we didn't talk very much about. We talked about NK cells, and now we're going to really be specifically focused on B cells and T cells, the mediators of adaptive immunity. So there are really three broad arms of adaptive immunity. You've got B cells that make antibodies, and antibodies themselves are soluble effector molecules. And so this can remove extracellular pathogens. It can remove pathogens that will eventually be intracellular, but can only remove them in their extracellular phase. And then you've got CD4 and CD8 T cells. So CD4 T cells, I call them the quarterbacks of the immune system. They're not generally direct effectors. They can be, but their major role and their major contribution is to induce other host cell responses and organize inflammation in a rational way. So they're required for the activation and differentiation of CD8 cells and also of B cells. You cripple both of these arms of effector adaptive immunity if you cripple CD4 T cells, which is why HIV over the long haul is such a deleterious disease. And then CD8 T cells are also direct effectors, kind of like antibodies, and that they kill infected cells directly. So this is critical for intracellular pathogen removal, viruses and bacteria that get inside cells. The final full clearance of them usually requires CD8 T cells. And the thing that invoked the word adaptive to be used to describe these is that they have memory. They can remember a pathogen that they saw before, and they're highly specific for the pathogens that they've seen before. So we're going to start with B cells. So B cells activate and bind antigen, and then proliferate and make antibodies that are their effector molecules that will eventually clear the pathogen. But they require help from CD4 T cells, which are also called T helper cells, which simultaneously are giving help to CD8 T cells that can go out and kill infected cells. CD4 T cells secrete tons and tons of cytokines, and so they'll feed back on that innate immune system, and particularly on macrophages. This is going to be a critical interaction for malaria and for TB when we get to those discussions later. And so they're all intimately interconnected. They can all act a little bit independently of each other, but for full functionality, they need to be together. All three of these cell types, their natural environment is in the lymphatics, which includes the spleen and the lymph nodes, which are studied throughout your body. We often talk as kids about your lymph nodes being swollen in your throat. There are cervical lymph nodes in your throat, but there are lymph nodes in literally every tissue, draining every tissue and housing B cells and T cells proximally so that they can mount an effective immune response for any tissue. Okay, so we mentioned the danger hypothesis and pattern recognition for innate immunity. Decades before those hypotheses, McFarland-Burnett had proposed the clonal selection hypothesis. And this is really one of the great achievements, I think, of medicine and of immunology of the last 100 years or so. He won the Nobel Prize. What he did was he formed this hypothesis based on just observations of the fact that people don't seem to be able to get infected with the same thing twice. And so this was before we had any concept of the lymphocyte. So the lymphocyte had not been discovered yet. Certainly B cells and T cells hadn't been discovered yet. There was some notion of serological immunity, which is what antibodies mediate. But he formed this just based on the behavior of organisms in response to repeated challenge with pathogens. And so he started thinking about how such a system must develop. And he had the idea, so he used the word lymphocyte and came up with the idea that each lymphocyte must bear a single type of receptor with unique specificity that can recognize a particular pathogen, and that an interaction between that foreign molecule of that pathogen and that lymphocyte receptor leads to the activation of that lymphocyte. And then those differentiated effector cells from the activated lymphocyte will each have that same antigen receptor, and that antigen receptors that are specific for self have to get deleted at some point because otherwise you would have autoreactivity. And so all of these postulates have turned out to be pretty true with obviously some very important details. But really, it's a beautifully written piece of work. Same thing, he wrote it as sort of a treatise, purely hypothetical, based on almost no data, but really guided his research and the research of many other people that then were validated over several decades. Okay, so we'll start with how B cells fulfill this hypothesis. So you've got a normal naive B cell floating around. It's got this molecule on its surface, which is an antibody and is also something we call the B cell receptor. So those two are the same thing. When it's on the surface of a B cell, we call it the B cell receptor. When it's not on the surface of the B cell, we call it an antibody. The business end of this protein can bind directly to foreign pathogens in a way that we'll talk about in detail in a few slides. And when it does so, it gets activated. Activation causes it to start a genetic program that will allow it to proliferate. All of its daughter cells have exactly the same antibody. So the antibody that saw the antigen gets expanded. The antibody that can't see the antigen does not get expanded. And eventually, these are going to become soluble and serve as the effectors of the immune response. So the question arose very early on after this proposal and after people were observing the fact that it seemed like we were able to make immune responses against any pathogen and actually against synthetic molecules that we had never even seen before. And the question was, how do you make enough antibodies or T cell receptors to be able to see all of these different pathogens? And a very important experiment was done in the 70s that showed that uniquely among cell types in the body, B cells and T cells actually changed their genome after development. And so this is, the classes, as we teach this, just keep getting younger and younger, and so people are less and less familiar with what this is. This is actually a southern blot. And so what it is you chop up DNA into semi-random fragments using restriction enzymes that cut in the same spot using a recognition sequence on DNA. And then you probe with different parts of that genome, little pieces of DNA like a primer, and that primer is radioactive, and it will light up a homologous region of DNA that you have on the membrane. And so this is a region of the B cell receptor in, say, a liver cell. And this is where it lights up on this gel. And this is where the V region of the B cell receptor lights up on this gel. And what they found is if they took a group of B cells, and these happen to be identical B cells, and they probed with the same probes, they were in different spots. And so what this meant was that in the B cells, this region of the genome had been rearranged. And so the restriction enzyme was cutting in different places. This also went on to win the Nobel Prize, just this gel. And so what has come out of that, what has come out of this is an understanding of what the antibody really is. And so this is structurally what it is. It is a dimer. So you always have two copies of what we call the heavy chain here. This is the heavy chain, so with 4 blocks here, and the light chain. And so this dimer of heavy and light chain makes one antigen recognition portion of an antibody, and there's always at least 2 copies of them. And this N-terminus is the hypervariable region, and it's actually, the hypervariable region is really in the middle of the protein, and this is what recognizes a foreign antigen. And the T cell receptor is going to have a very similar structure when we get to it. And so how does the DNA actually get cut up, and how do we generate all of that diversity that we can see so many different things with our antibodies? And so this is done via a process called VDJ recombination. And again, this is going to be replicated on the T cell side, so we'll just go through it here in detail. So it turns out that the B cell locus and the T cell locus have this very odd structure, and that they have panels of genes. So they've got a panel of B genes and a panel of J genes, and also a panel of D genes on the heavy chain. And that's what makes it heavy, is it has this extra gene segment. And when the B cell receptor is getting made, I'm going to actually show you here, this is how many different V genes there are, and how many different J genes there are, and how many different D genes there are. when in an individual cell, when it decides to make a B cell receptor, it picks just one of each of these genes, and it cuts the genome and fuses those gene segments together. And this is this process called VDJ recombination. And so first on the heavy chain, it'll pick a D region and one J region out of all of these D regions and all of these J regions, and it'll stitch them together, and everything that falls in between the D and the J that were actually picked, so let's say it picked this D and this J, all of these in the middle get cut out, and these two get fused together. And so these float off, they're not used anymore, and then that D and that J fusion will get fused to one of these V's. Let's say it picks this V, it'll cut out all of these V's in this little bracket here, and you'll get a V, a D, and a J, and then you'll have this big intron, and you actually don't fuse with the C. All of the rest of that is done by splicing. And so then the same thing happens on the light chain. And so you have this process of what we call somatic recombination. You're cutting up the genome, and you're stitching it back together. And you're doing this actually in a purposefully error-prone way. So at that fusion where the V, the D, and the J, or the V and the J here come together, you're on purpose making lots of mistakes that get repaired in sort of a sloppy way. And the whole point of that is to actually generate more and more diversity. And then that's going to be used to make the antibody. So this is actually even a little bit out of date. It's from 2005. But these are the number of unique light chain variable and heavy chain variable D and J regions. And together they can form an enormous diversity. On the low end, somewhere around 10 to the 15th. That 10 to the 15th number, which is what's still cited in Janeway, which is where this figure is from, was really derived in the sort of the late 80s, early 90s, based on the sequence data we had available then. A more recent estimate of diversity puts it in the range of potentially 10 to the, say, 50 to 60 unique potential BCRs or TCRs that can be generated. So that is an enormous number that is actually obviously never fully sampled in any individual human being or even in the mass of humanity. And so that just shows the numbers here. Again, these are generally underestimates, but even at these numbers, these are much higher numbers than the number of cells you ever generate in the course of your life. So maybe you have 10 to the 12th T cells in a human. And so there's at least 1 million times more potential T cells than T cells that we actually have. Okay, antibodies, unlike TCRs, one of their unique features is that they have different subclasses, and the subclasses relate to the constant region of the antibody that's being used. And so these are the subclasses: IgM, IgG, IgD, IgE, IgA. And the subclasses have different functional roles. And again, this is the constant region. So this is the business end of the antibody that binds the antigen. And the constant region down here is going to be the part of the antibody that can mediate certain effector functions and recruit diverse cell types. And so each of the different classes of antibody play sort of a different role. IgM is the first class that gets used. And so it is always secreted as a pentamer, so 5 copies of this molecule, 10 antigen binding domains, because the first antibodies that get made are probably not that great, and so you have to increase their valency to make them work a little bit better. And then as the antibody response matures, it'll class switch, so the same antibody is being put on a different class to invoke different effector mechanisms. And so the IgE antibody that can get secreted is very good at cross-linking receptors on the surface of mast cells and causing mast cells to degranulate. And that's critical in dealing with particularly worm infections. This is also what happens when you have allergies. These granules in the mast cells are full of histamine. And so when IgE binds to an allergen, it releases the histamine, and that's why you take an antihistamine. IgA is critical for mucosal surfaces, and IgA is always secreted as a dimer, so you get 2 of these IgA molecules secreted together, and they're joined by a special protein that links IgA molecules. And that special protein allows them to traverse epithelial surfaces and get secreted into mucosal sites. And so you have very high concentrations of IgA in your lungs and in your gut. And so these are really important for controlling the microbiome at those two surfaces. So another very unique feature of B cells and antibodies, even against compared to T cells, is that they get better with time. And so they continually mutate their antibody receptors with small point mutations, so that as you recall that response, it gets more and more avid binding to that antigen. And so it does this in a special structure in the lymph node called a germinal center, where the B cell sits next to a CD4 T cell, and it keeps some antigen around. So you keep a small amount of the pathogen around in your lymph node, and the B cell antibodies are binding to it as a sort of answer key that this is what we want to bind to, and we want to get better at binding to this. And it'll start making random mutations in the B cell receptor to try to get better. Now, most of those are going to make it worse, and if they make it worse, then the cell dies. But if it makes it better, then the cell lives and it gets to proliferate a little bit, and it'll get a signal to keep trying to keep getting better. And so you can get orders of magnitude increases in antibody-antigen affinity through cycles of this affinity maturation process. And during this process, not surprisingly, you don't want to secrete antibody because you might make an antibody that's self-reactive, or that's just totally useless. It's a waste of resources. And so it's all surface-bound antibody signaling to the B cell to either keep making mutations or stop. This summarizes those classes of antibodies that I told you about, and then how those different classes mediate different effects. So the dominant antibody type that gets made, that gets measured, it's everything that's being measured right now for coronavirus, is IgG. It's secreted in large amounts in the blood. And really, its main mechanism of attack is to just bind the pathogen and block whatever the pathogen's trying to do. Block the receptor from binding, the pathogen receptor from binding its receptor on the cell surface. Antibodies can activate complement, as I mentioned in the innate section, and so they can cause complement molecules to come flying down onto a bacterial surface and blow the bacteria up. And then they can also mediate localized changes in inflammation and in cell activation, like I showed you with the mast cell. Okay, I don't think I can do T cells in 5 minutes. We're probably going to run slightly over, but I will do it relatively quickly. And that's in part because we can do T cell receptors relatively quickly because they're just like B cell receptors, except that they're not secreted. And so they're solely membrane bound, and they're restricted in the type of antigens that they recognize. So B cells bind just proteins, sugars, anything on the surface of a pathogen. They are shapeshifters around that particular pathogen. T cells are what we are called restricted. So this is the T cell receptor, and it only sees antigen in the context of another molecule. And those molecules are all called major histocompatibility complex molecules. There are some very rare exceptions to this, and we're just not going to get into it in the course of this introduction, but those of you that are deep in immunology may have questions about that, and we can talk about it. But so the T cell receptor sees this MHC molecule, those molecules that the NK cells are surveilling for. And so that's a piece of self. And then it sees a little piece of the pathogen, a little peptide, usually somewhere between 8 amino acids and 20 amino acids. And that's it. The T cell only sees that little piece of the pathogen, and it only understands it in the context of this other molecule. And we call this MHC restriction. So the TCR binds both of those molecules. So briefly on the MHC is basically a box that holds peptides from pathogens and from self molecules as well. So there's cell surface receptors that contain this peptide binding cleft. This is looking straight down on the MHC, and then this is looking sideways at the MHC with the peptide binding cleft right there. And these small peptides get placed in there as a regular process by the cell. So they're constantly chewing up all the proteins that get made inside the cell, whether they're made by pathogens or whether they're made by self, and loading pieces of them into the MHC. This is just some crystal structures of what that looks like, looking straight down on the MHC here. And then what determines whether or not a cell is a CD4 cell or a CD8 cell is whether or not its T cell receptor sees antigen in the context of MHC class 1 or MHC class 2. And so these are these two big types of MHC molecules. And if you see antigen in the context of MHC class 2, you're a CD4 cell, and CD4 actually binds MHC class 2 to help stabilize that whole interaction. And similarly, CD8 binds MHC class 1 and stabilizes the TCR MHC peptide interaction. And so if you're a CD8 cell, you bind class 1 and you've got the CD8 molecule. If you're a CD4 cell, you bind class 2 with your CD4 molecule and your T cell receptor. Okay. So the last postulate of the clonal selection hypothesis was there had to be a way of getting rid of autoreactive cells. And for T cells, this is done in the thymus, where T cells learn whether or not they're better as an MHC class 1 binder or an MHC class 2 binder. And so that's where they decide if they're a CD4 or a CD8 cell. And then as part of that education process, they also decide whether or not they're seeing too much self-antigen and if they see it too well. And so in the thymus, which is where the T cell is developing, the TCR will scan dendritic cells and other antigen-presenting cells that have lots of MHC on their cell surface. And if they get a positive signal, they'll decide to live. So they need to see some MHC to live, and they decide whether or not they're a CD4 or CD8 T cell. But if they get too strong of a signal, that triggers this process called negative selection that causes them to kill themselves and die. So that you remove these cells that see self-antigen from the population extremely well. Again, I mentioned this, we've mentioned this cell type a few times. In the thymus here, this is dendritic cells. And these are an innate immune cell that are all throughout the body and are really the most efficient antigen presenting cells. So the most efficient cells at showing antigens to T cells in the context of MHC of any cell type in the body. And so these are generally required for the initiation of adaptive immunity. And so these cells will live, and they look very dendritic, which is how they got their name. These cells will live all throughout your body and will get activated by those innate immune signals that we talked about at the very beginning to go to the lymph nodes when they've picked up antigen and activate T cell responses by presenting antigens on peptide MHC. And so this is what that looks like. Again, you've got an antigen presenting cell, a dendritic cell or a macrophage. They've taken up an antigen, they've chopped it up into little peptides, they presented it on their MHC molecule, and they've now gone to find a cell with a T cell receptor that can uniquely see this class 2 molecule, the peptide. This is a class 2, so it's using CD4 to stabilize it, and it will activate that cell. Other cytokine signals and receptor-based signals from the antigen-presenting cell to the T cell will also occur, and so you'll hear people write about co-stimulation, and that will activate the naive T cell, will serve as that same activation that B cells get when they bind their antigen, that will cause it to proliferate and replicate this unique T cell receptor that can see the pathogen. This summarizes those different types of dendritic cells. So dendritic cells are really good at naive T cell presentation with high MHC expression once they get activated. They're ubiquitous throughout the body, and they migrate to the lymph node and deliver really large amounts of this co-stimulation. Macrophages also can do this, but they tend to live more in specific tissues. And then B cells, surprisingly, we've been talking about B cells as effector cells of the class 2 system, of the adaptive immune system. They actually have a lot of class 2 on their surface and can present very well to CD4 T cells. And so they can act actually as antigen presenting cells for CD4 T cells directly. And that's maybe not surprising because they have a very good way of getting antigens into their cells to process and present on class 2 to T cells because they can use their antibodies to get those antigens. And so there's a lot of pairing between B cells and CD4 T cells looking at the same antigens, one with an antibody and one with their T cell receptor. So again, all of this initial activation for T cells happens in the lymph node, and a lot of the initial activation for B cells will happen here too. And when you want a B cell and a CD4 T cell to form one of those germinal centers where they're going to talk to each other, the CD4 T cell is going to help the B cell affinity mature by talking to it and giving it cytokines and signals to change its B cell receptor. The B cell might present antigens to the CD4 T cell. There are these regions of the lymph node where all the naive B cells hang out, and there's this region of the lymph node where all the naive T cells hang out. And as T cells get activated and see a specific antigen, they'll leave all these other naive T cells that are not specific for that antigen behind, and they'll start migrating towards the B cells. And the B cells that saw a specific antigen will leave all the other B cells behind that didn't see that antigen, and they'll start migrating toward the T cells, and they'll meet in the middle. And they live right at that margin, and that's where those germinal centers form, where the B cells and T cells can talk to each other, but they leave everybody else sort of intact. So you have these really structured regions of the lymph node. Okay, so we've been talking about this class 1, class 2, CD8, CD4. I mentioned already that the CD8s are the killers, and it's important that you understand that they see antigen in the context of class 1, and that makes them effective killers, because MHC class 1 is present on every cell type in your body. So every cell expresses MHC class 1, and that's what makes it such a good target for NK cells to look for, because every cell should have it, because every cell is constantly chopping up little pieces of the proteins that it's making, presenting it on class 1, and making it available for surveillance by circulating CD8 T cells, which we also call cytotoxic T cells because they kill infected cells. So if a CD8 T cell that's been activated and licensed goes to the site of inflammation, following those chemokine and cytokine gradients, it gets into the tissue, it sees its antigen now in the context of MHC class 1, this little peptide that's from a pathogen, it will kill the targeted cell. Hopefully, any CD8 T cell that sees a self-antigen in the context of MHC class 1 didn't even make it out of the thymus, and so we don't have that problem. CD4 T cells really just have to talk to B cells, talk to macrophages, organize things. They're clearly in management. So they don't necessarily even have to go to the site of inflammation, and they only see things in the context of class 2. And class 2 is restricted only on professional antigen-presenting cells. So there's 3 cell types I showed you: B cells, dendritic cells, macrophages. And so if they go to the lung and they look at the lung epithelial cell, it's not going to have class 2 for the CD4 T cell to kill. There are exceptions where epithelial cells will upregulate class 2, but they're the exception, not the rule. So the CD4 T cell really stays talking to the professional components of the immune system. And it will talk to macrophages, it will give it cytokine signals, it'll tell it to kill bacteria more, make more reactive oxygen intermediates, really ramp this up. We're not getting rid of the infection. I can still see my antigen. You've got to get more and more inflammatory, take the brakes off. And it can talk to B cells and say, we've still got this giant worm around. We need you to make more antibodies that can help get rid of the worm. And you need to make better antibodies. And so I'm going to help you affinity mature. And so what CD4 T cells tell different cells to do depends on which type of CD4 T cell it is. And so this brings back this idea of type 1 T cells. So we got these Th1 cells. These are going to help CD8 T cells. These are going to promote the production of killer responses, killing infected cells, intracellular pathogens, viruses, and bacteria. And the signature cytokine that CD4 T cells are going to make to do this is interferon gamma. And so we call that interferon gamma as part of the Th1 response, or type 1 response, T helper 1 type 1 response. And so we saw this a little bit very briefly with the innate lymphoid cells. It's the same type 1 response. The type 2 response, more associated with antibodies, large extracellular pathogens, tissue reorganization, allergy and asthma. The critical cytokine here is IL-4. There are many other cytokines associated with each of these, but just for the sake of time and simplicity. So we've got interferon gamma, type 1 responses, IL-4, type 2 responses. And then IL-17 are those weird extracellular, intracellular bacteria, mucosal infections. Annoyingly, we call this the Th17 response, but it's the type 3 response that I showed you earlier. So type 3 and Th17 are identical in the literature. Okay, so we've done all of this work in the lymph node. We've licensed CD4 or CD8 T cells to go out and kill an infected cell. So it initially needs that licensing from a dendritic cell. It'll leave the lymph node. It'll go to the lung or the skin or the gut or wherever the infection is. It will see its antigen again on a cell surface, and it will kill the cell. And it'll kill the cell using a lot of those same mechanisms that complement uses. It'll poke a ton of holes in the cells, or that NK cells use. And again, it's convergent evolution. It's not the same as complement. It is the same as the NK cell. They're using the same effector molecules, these little bombs that go in and blow up the cell. Can also kill the cell through just telling the cell to kill itself. There are receptors on the surfaces of cells that if the CD8 T cell ligates it, it'll say, you know what, you need to kill yourself. And the epithelial cell will say, okay, I'm infected, I'm going to kill myself now. This just summarizes everything I've just told you. So I just put it in here for your reference. CD8 T cells are the killers. CD4 T cells are the helpers, so they can determine whether it's a type 1 response to kill intracellular pathogens, a type 2 response to get rid of large extracellular pathogens. You can have elements of both responses occurring at the same time. I'm almost done here. This just shows kind of echoing back that first slide I showed you where we have these really different types of pathogens we're faced with, different sizes, and that requires really different types of effector mechanisms, whether CD8 T cells are involved or whether or not the determined by whether or not the pathogen is small enough to fit inside a cell. If it doesn't fit inside a cell, then there's no point for CD8 T cells because they can only kill host cells that are infected with something. If it's really big, then antibodies are going to be important. But even if it's really small, antibodies are important because they can prevent a virus from getting from one cell to the next cell. Or if you're vaccinated, they can prevent the virus from ever getting into a cell at all. So the last question is, when does the immune system shut off? And so this is regulated by specialized cells that talk either via direct contact or are a different set of secreted cytokines. And then there are lots of inhibition loops. I always said there's a stop for every go signal that's activated in the immune system. And if that fails, then that's how you get autoimmunity, and that's actually how you get asthma and allergy as well. And the main cell type that is studied for this purpose, that there are many effector mechanisms of regulation, is the cell type called the Treg. And this is another type of CD4 T cell, but it's a CD4 T cell that is expressing a particular transcription factor called FOXP3, and it gets generated in the thymus, and it can also be generated in the periphery. And one of the ways that it gets generated is it sees its antigen too well. And so if it sees its antigen too well in the thymus, I told you before, then it should delete itself. Well, a subset of those cells don't actually delete themselves. They turn on this FOXP3 transcription factor, and instead of going around and killing self molecules or self molecule bearing cells, they'll go around, and if they see a lot of self molecules, they'll associate this with damage, and they'll try to shut off the surrounding immune response. And so they are sentinels of tissue damage in the periphery. And then cells can get induced in the periphery if there's just too much inflammation around. CD4 T cells will start converting into this regulatory phenotype, again with the expression of FOXP3, and they'll try to tamp everything down. And the mechanisms by which they do this are actually still an incredibly hot area of study in immunology. I don't think we really fully understand how they work, but we do know that if you don't have Tregs, you will die of massive systemic autoimmunity, and there's nothing that can be done to stop it except a transplant. These are some of the mechanisms that Tregs can use. They can actually directly kill other immune cells, so sort of fratricide within the immune system to try to get it to shut off. They can secrete lots of cytokines and target other aspects of the immune system to try to calm them down. And then the very, very last thing is you've now successfully calmed the immune response down, you've contracted, you've cleared the pathogen. How are you now protected from subsequent exposures of that pathogen? And that's through the generation of T cell and B cell memory, which is unique to the adaptive immune system. And so your first infection, it can take over a week to start generating antibodies. With SARS-CoV-2, we're seeing now, people are sometimes not detectable for 2 or 3 weeks. But then if you get re-exposed again and these antibodies are good and effective, you might have an expansion of your adaptive immune effectors, but you won't even know that you're sick. So the virus will never get inside your cell. You'll expand and make lots more antibodies, and they'll clear out the infection almost instantaneously. Memory is not forever. It can last quite a long time, though. This is obviously something that's in the news a lot now, which is weird from an immunologist perspective to see people talking about T cell memory and B cell memory in the New York Times. But in general, CD8 T cell memory is generally very good if it's generated in a good vaccine or via infection, and can stay flat for your entire life. If you get the yellow fever vaccine, you will probably have yellow fever specific CD8 T cells until you die for decades. CD4 T cells tend to decline more over time, and this decline is not fully understood, and the mechanisms that cause it are not fully understood. Lots of interesting mathematical modeling around this, though. And similarly, B cells are thought to generally last a lifetime if generated under the right conditions with the right amount of CD4 T cell help. And that's critical, and that's where a lot of vaccines don't work well, is they don't generate good CD4 T cell help to promote good B cell memory. This is my last slide. Just briefly summarizes this innate adaptive divide, these weird cell types in the middle that bridge innate adaptive immunity. And I'm happy to take any questions. And again, there's lots of time later to go into more depth into any of these topics. And then we'll go into more depth on antiviral immunity and anti-complex pathogen immunity tomorrow.